welcome to the YouTube channel of Independent Science News. My name is Jonathan Latham and I am the editor of that website. So in the last video I talked about how we've written a scientific paper which is kind of a re-evaluation of the toxicity of Bt proteins. And Bt proteins are insecticides that GMO companies, Monsanto, Syngenta and so forth, put into GMO crops. And these are insecticides, and we wanted to investigate better the toxicity of those proteins, right? And see whether they really were similar to the proteins used by organic farmers that you always hear from the PR people, and that this is the standard industry talking point, is that these are basically proteins used by organic farmers. And what we did is show that that's not true, right? That there are all these differences, and these differences have important toxicological implications, some of which have been investigated and some of which have not. So, but what was also in that paper, which is really an interesting aspect to it, is when we were looking at the regulatory documents that companies were supplying, it was obvious that there was a high level of deceit going on here in terms of the data and information provided by the regulators. In terms of the data provided to the regulators by the companies. Let's talk about some of those examples. So the first example is that the companies are testing. Instead of they're doing tests, for example, on soil degradation, or on they're feeding to rats, or they are feeding to non-target insects. They're basically doing a whole suite of risk assessment experiments, and they're using what they call surrogate proteins. So these surrogate proteins are not the proteins that you will be eating when you ingest a GMO crop, right? That was one of the findings of our paper. But anybody could tell you that, because these proteins are different from the proteins produced in a GMO corn, for example. Like the protein produced in a GMO corn is wrapped inside a cell, right? So it won't necessarily biodegrade when it passes through your stomach, for example. And so there are in theory many other differences between these surrogate proteins and the proteins that people will actually be eating, that organisms will actually be eating, that will enter the soil, and so forth. But so the use of these surrogate proteins is a problematic aspect of the risk assessment, which has been attention has been drawn to this multiple times. Regulators never do anything about it, and the companies keep supplying surrogate proteins. But then what happens is sometimes those surrogate proteins, which you can call fake proteins if you like, because they're not the thing that you'll be eating, they're even put into fake experiments. So for example, regulators, or the companies rather, will take those proteins and they will subject them to what they call in vitro digestion experiments. So these are digestion experiments meant to simulate the human gut, and to hopefully demonstrate, as far as the regulators and the companies see it, to hopefully demonstrate that these proteins are fully degraded when they pass through the human gut. Now, firstly, this is a surrogate protein being digested in a surrogate gut. That means that experiment is not realistic. Another aspect of that experiment is the definition of degradation of those proteins inside the gut, right? These are not tests where they're showing that the whole protein is disappearing. They're just showing that the protein they put in there is not the same as the protein that they took out, right? So they have basically an antibody test which they're using to detect that protein, and if the protein is slightly altered, then it will not be detected by the antibody test, and therefore the protein will be defined as having disappeared in that test. So you've got essentially an unrealistic test of what actually is degradation, because as I pointed out in the previous video, one of the interesting things about these proteins is that the smaller they are, the more toxic they are, right? The biodegradation, by that definition, is totally a different thing from actually making that protein safe. So you've got basically fake proteins being put into fake experiments and subjected to fake biodegradation assays, right? So this is part of the problem. The other part of the problem is that the risk assessment, when you start to use surrogate proteins, the risk assessment becomes a exercise in trying to show that the surrogate protein is the same as the protein that's produced inside the GMO crop, right? So that becomes a huge focus of the risk assessment, and this none of which experiment would be necessary if the companies actually use the real proteins from the crop or the crop tissue itself to do their experiments on, right? If they were doing realistic experiments, they wouldn't have to spend all this time demonstrating that surrogate protein and the real protein were the same as each other. So they end up doing all these experiments to supposedly show that the surrogate protein is the same in size and has all these biochemical properties that are the same as the natural protein or the natural GMO protein. And what they do to test that is they basically extract the natural protein from the crop, and then they look to see, they run it on a gel, on a size selecting gel, and try to show that the GMO protein is the same as the surrogate. But before they've done that, essentially what they've had to do is purify it, because the protein is relatively rare inside a plant cell. They've had to purify it, and the method that they use to purify these proteins actually selects away from basically unusual cryoproteins, which is the whole point of the assay, is to find out whether there are such things. The assay actually selects against these unusual cryoproteins, because the companies are doing things like they're digesting the extract, right? They are running it over a column that loses, every aspect of a scientist will understand this. Every aspect of a scientific experiment that essentially results in losses, right? And some of the losses of information are extremely large, right? If you run a protein over a column and the column only selects for certain size proteins, basically all proteins of unusual sizes will be lost, right? And so the companies are using whole combinations of assays in these purification experiments, and then they present side-by-side analysis of the surrogate and the plant-produced protein. But the plant-produced protein has been subjected to all these analyses that essentially select against unusual size proteins being found. But they're not explaining this to the regulators, right? I don't think the regulators have any understanding of what's going on in these experiments. And if they do, they're not, they really ought to be asking difficult questions of these experiments. But basically, by running them over columns, by selecting the proteins with antibodies, by sometimes selecting with antibodies to other ends of the cryoproteins, essentially what is happening is that proteins that don't fit the official version of what a cryoprotein should look like are being lost from these assays. And that essentially is fraudulent research, right? And so this has, because the whole purpose of the assay has moved from working out whether the GMO crop is going to actually harm non-target organisms, whether it's going to harm people, whether it's going to harm livestock. The whole purpose of the risk assessment has become this attempt to demonstrate that the surrogate and the GMO proteins are the same as each other, and then those experiments are being done fraudulently. What you end up is essentially a fraudulent risk assessment. And so we explain all this in the paper. And so we show how all the different assays in combination and individually are actually selecting for the kind of things that the companies want to demonstrate and against the kinds of things that would cause concern to the regulators. But there appears to be no appreciation by the regulators of what is actually going on here, and they're basically being hoodwinked by the companies. So I wanted to explain that, because we've never written about that aspect of our paper, and so I wanted to describe it on this channel episode, so that people would read the paper a little bit more carefully and try to understand that aspect of it, because we emphasize in the title how GMO and and naturally occurring cryoproteins were different from each other. But this whole issue becomes, you know, the sort of central aspect in many ways of the risk assessment process. And so you know, even though this is not the conventional subject of a scientific paper, it is a really interesting phenomenon from the point of view of protecting public health, right? But also more generically about the relationship between regulators and applicants when it comes to the kind of quality of data and the inherent biases of that data that are presented to them. So, well, thanks very much for watching, and hopefully we'll see you next week.